Thank you very much to Alma to give me and my laboratory the opportunity to talk about what kind of dictates our life. And this is bovine spongiform encephalopathy. When I was preparing this presentation, I thought I'd break it down into a couple of different parts. First of all, I'll give you a very short introduction into those enigmatic and miasmatic diseases. BSE, or bovine spongiform encephalopathy, is part of it, and its human counterpart, variant Creutzfeldt-Jakob disease. I'm also going to talk about the situation in this 2015 and why we still care about it or need to care about it. And finally, I'm briefly talking about research done in BSE, and as it happens to be mainly in Lethbridge, not because we are so brilliant, we are brilliant of course, but mainly because not a lot of people do BSE research anymore, left alone doing research by infecting cattle as we do. So, okay, where are we? Spongiform encephalopathies is a group of diseases which is characterized by neurological signs, and they affect both humans and animals alike, and it is mainly dementia and ataxia which is presented. And the brain is the target. The brain actually is the only target of those diseases. And in comparison to other infectious diseases, the incubation time, the time until disease shows, is super long in those. We are talking years as compared to weeks, or sometimes even in food poisoning, minutes, when you are lucky to reach the next bathroom before everything is starting to happen. The definitive diagnosis of these diseases are always only happening on brain, meaning it only occurs at necropsy, we can't say before. And they are either of genetic origin, very much in humans. They are happening spontaneously, so we think at least. And they can be of infectious nature. And infection only occurs by one way, and this is by, fancy call it, dietary exposure. That means either by feeding contaminated feedstuff to animals or to eating contaminated food products in the case of humans. Of course, we have the experimental infection, that's what we do in the laboratory. And then there are those unfortunate humans which have been accidentally infected by not enough decontaminated instruments or by tainted blood products. This is a picture of the brain of our first BSE case found in 2003. And when you look at it, you appreciate why those diseases are called spongiform, because the brain really looks like a sponge. And the holes occur because infected brain cells drop out and die. In contrast to other infectious diseases, the infectious agent for spongiform encephalopathies is based on a normal protein which we all carry, all of us mammalians carry in the brain, in the spinal cord, in the spleen, lymph nodes, and in blood cells. And interestingly enough, although we know this cellular PRPC, cellular protein, since around 50 years, we still don't know very much what it is all about. We know for the brain it is important for maintenance of brain function and nerve cell functions, but we have no idea what it is doing in the periphery, in the spleen or in the lymph nodes. And what infection does, it induces a change in shape, and the change in shape induces a change in property. And the protein, which is normally soluble in the brain, becomes insoluble, clumps, falls out, and forms those amyloid deposits, and the brain gets choked up and cannot function anymore. For us, important in the laboratory, but also very important for farmer or the medical community, is the extreme resistance to decontamination of those agents. We do run our lab waste at 135 degrees Celsius for 30 minutes at 2 bar pressure in order to get rid of the prion protein. Normal viruses, for example, rabies virus, you have it on the outside on the bench for 10 minutes and it is already decontaminated. So quite different. This is, again, the brain of this first animal from 2003. You have in the center a very nice, still intact-looking brain cell, and with all its little ramifications, and where you see the brown color is where the prion protein is located. So where are we now? What do we know about bovine spongiform encephalopathy? Not talking about that it was detected first in the UK and so on and so forth. I'm talking about the important things, especially for us in Canada. It is a single animal disease. It's always less than 3% of the animals in one herd infected. That, by the way, makes our last case from spring 2015 so interesting and so devastating. And changes in the UK rendering induced the contamination of feedstuff with BSE, and due to feeding of the contaminated feedstuff, it was amplified in national herds, including the Canadian national herd. The youngest field case, animals which are infected by feedstuff, is 18 months of age, and this was detected in the United Kingdom. The oldest animal was around 19 years, and this is a case from France. This enormous difference in incubation time is related to the exposure, is related to the fact of how much or how little these animals had received. There is no shedding of infected prions by BSE, so there is no prions are in saliva, in urine or feces. That is very different to chronic wasting disease or scrapie. And there is no vertical transmission, meaning it is not transmitted from one herd animal to the next. It is really only by feeding, and there is also no horizontal transmission. There is no transmission from mother to offspring. All of this is very, very different to chronic wasting disease in cervix and moose, and to scrapie, the same disease in sheep and goat. So we know now there are different strains. Atypical BSE has been detected and characterized. There might be spontaneous forms. We don't know much about it. But what we know is that BSE is the cause of variant Creutzfeldt-Jakob disease, the human disease, and this is acknowledged and communicated as of March 1996. So where are we now? Now, and those are the latest numbers available, now in last year we had only 12 cases worldwide, in comparison to 1997, where only in the UK 38,000 animals were confirmed with BSE positive. So there is a dramatic decline in numbers. Canada has those 20 cases, 2 are atypical BSE, and 18 are classical, and this is as of June of this year. And I counted, because it is a Canadian case, the US case from 2003 into ours. There are the last 2 variant Creutzfeldt-Jakob cases were reported in 2012. There is no case in 2013 or 2014, and as of 2014, 229 cases of variant Creutzfeldt-Jakob disease are reported, and the vast majority of those, 175, are detected in the United Kingdom, in the country with the highest prevalence of BSE. So why do we still care when we have such a significant drop in numbers? And why do we still do research in it? The main reason is atypical BSE. And it's atypical because of the enormous imagination of the scientists, because it behaves atypical in one of our detection tests, which is a Western blot, segregating a protein according to its molecular weight. When you look at the very left lane in the box, you see those typical 3-band pattern, typical for prion disease, those bands. And when you look at the lowest band and focus on the lowest band and move to the lane in the middle, you see that this lowest lane is shifted a little bit lower. Hence, we called it L-type or lower BSE. And the sample on the very right corner is an H-type BSE case because it is shifted up a little bit higher. And this is how the nomenclature occurred. So atypical BSE is found worldwide in a very low number. There are only around 83 to 85 cases. It is found everywhere in the world, in Japan as well as in Europe, as well as in North and in South America. And there are countries with high numbers. France, for example, has a relative high number, or Poland for both types, and we don't really know much why this is the case. Then there is Ireland, for example, with only H-type BSE. And then there is Italy, for example, with only L-type BSE. Why that is the case, we don't really know at all. And then there are countries where atypical BSE is the only form of BSE detected. Sweden is one of those unfortunate countries with its very robust surveillance system. And then it's supposedly to be the same in the United States as well as in Brazil. So what do we need to deal with, or why do we need to look at atypical BSE from a regulatory point of view at least, if not from scientific as well? We need to find out if atypical BSE can be transmitted into cattle by feeding. If this is the case, then we are in the same situation as we had been with classical BSE. We also need to know if the specified risk material, SRM, in atypical BSE is identical to classical BSE, and we don't know that yet. And while we are at it, infecting cattle, we thought we also want to finally answer the question if only calves, animals at around 6 months of age, can become infected with BSE, or animals at an older age as well. And what we have done, and this is thanks to ALMA and other provincial funding sources, we fed two different groups of cattle with H-type, L-type, and C-type BSE. One group was around 3 months of age, and the other group was 14 months of age. And then we examine for clinical signs, and we sample monthly and euthanize, and then look at tissue. This is the experimental schedule. We have this group of 7 calves, and out of the group of H-type BSE, 1 animal had to be euthanized, and out of the group of L-type BSE, 1 of the animals had to be euthanized as well. And when you look at the yearlings infected, all animals with atypical BSE are still alive and doing well. Only the animals infected with classical BSE as adults all had to be euthanized. And since the clinical presentation is so important for our call when we need to kill an animal, I thought I'll quickly show you how the animals are behaving differently. It's a monthly procedure. They all know the drill over the years, and just look and see how different these animals respond to sound. This is a normal control animal. And to light now, the flashlight is pretty much bored and couldn't care less. And then to touch. So it's pretty non-invasive. And then we, of course, monitor movement and behavior once they go back into the pens. So taking all the sweet time, checking here and there, and had to be, let's move a bit. Okay, and now the infected animals, please. So this is classical BSE. So it's quite intense, the reaction to sound. And even more to light. And it's getting a little, starting to show problems in movement. It's a little bit stiff, and that was starting to develop. This is now H-type BSE.
The tremor? Yeah.
These animals know this since years, this procedure, and see how different. Lots of salivation, and this is L-type, the most pronounced. They never settle down.
It's time.
So, not so much involvement of the motor system in L-type BSE, but this significant change in behavior. And that was true for all of them. So what are those animals, what are the results of those animals? We had to put down the one animal with L-type BSE, fed. We euthanized 19 months after challenge because of progressive clinical signs. And most interestingly for us, it was negative by all of our BSE standard tests, including the super sensitive test methods we have available. And so far, we cannot detect any infectious prion protein in this animal at all. And what we have done as a consequence of this, we have again infected animals, this time by intracranial challenge, to find out if this behavior, this change in behavior was really related to L-type BSE. The difference is in H-type BSE, this one animal which was euthanized even earlier with 17 months, again with progressive clinical disease, was initially negative, but when we did more enhanced and super sensitive test methods, we found PRP-RS or prion protein in the brain of this animal. So that means there is some indication that atypical BSE can actually transmit it by feeding. And important for us to remember and to know is that this was in an area of the brain which is usually not involved in classical BSE at all. And again, this animal, brain of this animal had been passaged on in cattle to detect further infectivity. So those three animals which were infected as adults with classical BSE, they all developed very progressive clinical disease. We euthanized them at 53 months of challenge, and 2 out of those 3 animals were strongly positive in the brain in all the areas we were expecting it. but one animal was not. Although, again, it showed clinical disease, it was negative by our standard tests, and it was only positive by our super sensitive methods. And in addition, we also had a very uncommon and unusual location of the signal in the brain by one of our tests, very similar to one of the human prion diseases, sporadic Creutzfeldt-Jakob disease. And we are currently investigating further what this might mean and what the implication for control of the disease might be. This cartoon shows you only one information which is really interesting and important in this scenario is the pedigree, because we have this very closed line-bred, almost inbred herd in our lab, and we are using for our experiments, and we could identify that those animals which were strongly infected and highly susceptible for classical BSE were very closely related to each other via the paternal as well as the maternal side. And the third animal, which showed a relative resistance to BSE, was the one which was least related to each other. So there seems to be an indication that there is something in the genetic susceptibility of cattle as well. And we have not known that before, and we had no indication for that before. So as results and conclusions, BSE and chronic and variant Creutzfeldt-Jakob disease numbers continue to decline worldwide, and that is the consequence of the regulation and the feed bans worldwide. The problem for Canada is, with our last case of this year, the application to the OIE, the World Animal Health Organization, for a negligible risk categorization is pushed back until 2020. It's always 11 years after the birth year of the last case, and the case of this year was born in 2009. But even with a negligible risk status, we all need to keep our BSE surveillance up and also all our other control measures. For the research data, H-type BSE induces in 1 out of 3 animals clinical disease, and we have PRP, or the prion protein, detected in the brain. Not so much for L-type BSE, but there are still 2 animals incubating, or we were waiting for those animals. And until further data available, we need to be we need to take a precautionary approach to atypical BSE as well as to classical BSE, and the OIE is starting to acknowledge this and potentially revising their approach. C-type BSE can infect adult cattle as well, which has an impact on our future disease investigations. It's then not only the birth farm we need to look for trace-out animals, but any other subsequent farm the animal was on after. We have indirect evidence, actually now even a bit more direct evidence for genetic resistance, and this is the first time that this has been found and reported for BSE, and we do further research in this scenario. I want to acknowledge my people in the laboratory. All of us are working hard and diligently. They are a cheerful and happy bunch, makes it's a lot of pleasure to working with them. I want to acknowledge my collaborators at the University of Calgary. Michel Levy, you saw him doing the neurological examination, and at Pager, and my friends and counterparts at the German Reference Laboratory. And many thanks to the great support of the province via ALMA, via the Alberta Prion Research Institute, and Health Canada, and lately also by the CFIA. Thank you very much.
Sorry, Stephanie. Thank you, Stephanie. We do have time for one quick question, if
Could you flesh out more of the information on the genetic connection? And I think it highlights the importance of maintaining this herd where you can actually make these types of discoveries. And you might comment, it's maybe the only herd in the world right now, so we cannot lose that.
Yeah. The CFIA, it's a mixed Hereford-Angus cross herd and maintained since the 1960s. It is new genetics are only introduced via artificial insemination. We also have finishing bulls. The herd is around 150 heads of animals, and it's at least line bred, if not inbred. And we didn't really know what important tool we had in our hands until we got these results. And you are totally correct. When you do BSE research, not that many people are doing it anymore, but those in Italy or in Germany, they just purchase animals from somewhere, not knowing their genetic background, not knowing their connection to each other. And those 3 C-type animals, they were half-brothers via the paternal side, and this is the bull called like Magic Johnson or something like that. We have the information, and the 2 susceptible ones are also very closely related via the maternal side. So the only difference between those 3 animals is really the father from the maternal side from this one animal. And we know the father. We know. We hope that there is still semen available, because we want to do some genetic testing, since it seems there is some genetic deletion in this one animal, and we want to make sure that we can trace back where this might be coming from. We also have offspring and relatives of this animal in the herd, and we are currently doing the genetics of those ones as well. So maybe there are cattle really resistant to BSE. This is the first time that we have some information.